Welcome to the Deep Dive. We sift through the latest research for you, bringing out those insights, those surprising facts. And today, well, we're diving into something that really shifts our understanding of a plant. Many of us think we know pretty well, cannabis. We're talking about new science, revealing a sort of hidden layer of chemical complexity. It proves there are still these extraordinary treasures, waiting to be found, even in the parts of the plant we often just, well, dismiss as waste.
And that's exactly it. It's a perfect example, really, of how even things we've studied a lot, these natural sources, can still hold profound chemical secrets we haven't uncovered yet. Our deep dive today, it's all anchored in this remarkable piece of research. It's from chemist at Stellenbosch University, just published in the Journal of Chromatography A. It really shows how, persistence and these innovative techniques can just open up completely new areas of knowledge.
Okay, right. Let's unpack this then, because what they actually found is, well, it's pretty eye-opening. For the very first time, researchers have identified this rare group of phenolic compounds. They're called flavoalkaloids. And the real kicker, they found them mostly in cannabis leaves, which
Yeah.
is interesting because this isn't just another finding about cannabinoids, right? That's been the traditional focus for decades. The compounds like THC or CBD that affect mood. This discovery, it shines a bright, completely new light on a frankly overlooked part of the plant's amazing chemistry.
Yeah, the significance here, it really can't be overstated. Phenolic compounds generally, especially that broader category, flavonoids, they're already super valuable. We know this in the pharmaceutical field. They have well-documented antioxidant, anti-inflammatory, even anti-carcinogenic effects. You think about how many health supplements or medicines have plant phenols in them. But flavoalkaloids, they are an exceptionally rare subset of these compounds in nature. I mean, their complex structures just aren't commonly found. So finding them in cannabis isn't just notable, it's a truly significant finding. It basically blasts open entirely new research pathways.
Okay, so these flavoalkaloids are rare. Got it. How much do they actually find?
So the Stellenbosch team, they meticulously looked at 3 commercial cannabis strains right there in South Africa. And they identified, get this, 79 distinct phenolic compounds. Out of those, an astonishing 25 had never, ever been reported in cannabis before. And a remarkable 16 were tentatively classified as these elusive flavoalkaloids.
60.
Yeah, 16. And what's really important to note here is that these rare flavoalkaloids, they were found predominantly in the leaves of just one of the 3 strains they looked at, which really underscores, the huge genetic variation and the nuanced chemistry that exists even within the same plant species.
16 new flavoalkaloids plus another 9 phenolic compounds nobody had seen in cannabis before. That's a huge number of new rare molecules, all with potential medicinal uses, you're saying. But it really makes you wonder, if these compounds are so valuable, potentially, why did it take until now to find them? I mean, cannabis has been studied for ages, sometimes really intensively. Was it not a lack of interest, maybe, but maybe not having the right tools or perhaps looking in the wrong place?
You're absolutely hitting on the core challenge there. It definitely wasn't a lack of interest. It was an immense analytical hurdle. See, the difficulties in analyzing plant phenolics, they're considerable. They usually exist in really low concentrations in the plant itself. And they have extreme structural diversity, meaning they come in lots of different, often very subtly varied forms. Imagine trying to find one tiny specific grain of spice in a really, really complex soup.
Right, okay.
Dr. Margriet Milliner, she's an analytical chemist at Stellenbosch University's Central Analytical Facility, the study's first author. She's really highlighted this. She observed that cannabis itself is just extremely complex.
Yeah.
It's a plant with more than 750 different metabolites.
Yeah.
And just to clarify for you, metabolites are basically all the chemical compounds a plant makes as part of its life processes. So finding such high variation in these phenolic profiles between just 3 strains, like she mentioned, that was genuinely surprising even to the researchers. It really speaks to this hidden complexity that was always there, just waiting for the right tools to uncover it.
So yeah, it's like trying to find that specific microscopic grain of gold on a huge beach, but maybe you didn't have a high-powered metal detector before. You kind of know it might be there, but you just can't pinpoint it.
Okay, that makes sense. So what was this innovative method then? What allowed them to finally crack this puzzle? How did they find what was hidden basically in plain sight?
Precisely. Your gold on the beach analogy, that's spot on. It really came down to some brilliant methodological innovation. Dr. Margriet Muller, this was part of her postgraduate work, she designed these advanced analytical techniques. They integrate comprehensive 2-dimensional liquid chromatography with high-resolution mass spectrometry. Now, I know that's a mouthful, but let's break it down. Think of the chromatography part as like a super sieve for molecules, and the mass spectrometry is like an incredibly precise chemical fingerprint scanner that follows it.
Okay, so the two-dimensional liquid chromatography bit, that's the super sieve. It sifts things out not just once, but maybe twice from different angles or something. Exactly. Yeah, you've got it. It separates compounds with incredible precision, far, far beyond what traditional older methods can do. And then the high-resolution mass spectrometry, that's the super-sensitive scanner part. It identifies each tiny compound by its unique molecular weight and how it breaks apart, its fragmentation pattern. That gives it a definitive chemical fingerprint.
Okay. And did they develop this just for cannabis?
Well, no, and that's important context. These techniques weren't just, pulled out of thin air specifically for cannabis. Dr. Mueller refined these powerful methods, first proving they worked really well on other complex natural products, things like rooibos tea, grapes, wine. So that background was crucial. It showed the method was robust before they applied it to cannabis. And they chose cannabis specifically because, well, everyone knew it was complex, and there was this understanding that its phenolics just hadn't been properly characterized.
Right, so they tested the tools first.
Exactly. And Professor André de Villiers, who led the study, the main author, he emphasized that the superior performance of this two-dimensional liquid chromatography was absolutely key. It's what allowed them to separate these really rare flavoalkaloids from the much, much more abundant flavonoids, the ones that often overshadow everything else. That separation power enabled their unprecedented detection in cannabis. Without it, these compounds would have just stayed hidden in the chemical noise of the plant.
That makes so much sense. It really is about having the right tool for an incredibly subtle, delicate job. But let me just play devil's advocate for a second. How sure are they that these newly identified compounds are really unique and not just, tiny variations of something already known, especially given how complex you said the plant is?
That's a really important question, a fair one. And you'll notice the tentatively classified wording for some of the flavoalkaloids that reflects proper scientific caution. However, the high-resolution mass spectrometry, it provides extremely precise molecular formulas in those fragmentation patterns I mentioned. That gives chemists a very strong basis for identification. It's not just a guess. And the fact that 25 distinct phenolic compounds had never been reported in cannabis before, period, and that 16 were classified as these very, very rare flavoalkaloids, well, that strongly points to genuinely new discoveries. The researchers are extremely confident this isn't just background noise or slight variations. It's a significant expansion of our understanding of what's actually in cannabis.
Okay, that's reassuring. So stepping back then, what does this all mean for us, for you listening? Beyond the lab, beyond the cool science bit, what are the broader implications? Finding these rare things in cannabis leaves parts previously just seen as waste? What's the so what here?
Right. If we connect this to the bigger picture, this discovery is genuinely transformative for several reasons. Professor de Villiers, he explicitly concluded, and I'm quoting here, that this analysis again highlights the medicinal potential of cannabis plant material currently regarded as waste. So, this isn't just about finding new compounds, it's about potentially shifting the whole paradigm of how we view the plant itself. For so long, the focus, especially in medical or industrial uses, has been almost entirely on the buds, right, and the cannabinoids in them. But this research, it forces us, really, to challenge our perception of what parts of a plant are valuable versus what's just waste. Suddenly, those discarded leaves, which might have just been composted or thrown out, they're revealed as potentially being this treasure trove full of unique chemistry.
Okay, so a big shift in perception. But beyond that, what does it actually open up? In terms of real-world applications, I can imagine scientists are already buzzing about this. But for someone like me, are you listening? What might this mean down the line?
Absolutely. Fundamentally, this discovery opens up entirely new pathways for biomedical research. It's a big deal. It encourages a pretty significant shift in focus, moving beyond just the well-known, heavily studied cannabinoids, and directing attention to what Professor de Villiers called cannabis's rich and unique non-cannabinoid phenolic profile. So what that means is researchers can now start exploring the potential of these newly found flavoalkaloids, and the other phenolics too, for completely new applications, medicine, pharmacology, maybe even other industries.
Like what kind of applications?
Well, for instance, given what we know about phenolics, generally their antioxidant, anti-inflammatory properties, these specific new compounds could be investigated for treating conditions linked to, say, oxidative stress or chronic inflammation. Think about certain neurodegenerative diseases or metabolic disorders, maybe even some types of cancer. Imagine the potential for developing novel drugs, new therapeutic agents that use these previously unknown compounds. Maybe they'd work differently or have fewer side effects than what we have now.
It really is incredible to think about that potential. And I guess it's not just about medicine, is it? There are probably wider benefits, too.
Precisely. For you, the listener, it's definitely worth considering the profound economic and environmental benefits as well. That's a huge angle. We can actually transform what was once just discarded material, literally, plant waste, into a valuable source of rare, potent compounds. Well, that has immense implications. We could be talking about new pathways for sustainable agriculture, where every single part of the crop potentially has value, which would drastically reduce waste. It could even lead to creating entirely new bio-based industries, creating jobs, economic opportunities, all from something that was previously just a disposal problem. This really underlines that there is still much to gain from studying cannabis, as the researchers put it, and by extension, probably countless other natural botanical sources that we might think we already fully understand. It's a powerful reminder, isn't it, of nature's unending complexity and generosity, and also our own capacity for scientific ingenuity.
It's truly amazing. Yeah, this deep dive really has shown us that even plants we think we know inside out, they can still hold these extraordinary hidden chemical secrets, especially remarkably in the parts we might just casually dismiss or throw away. It's quite humbling, actually, a reminder of how much more there is to learn from the natural world right under our noses.
And it really raises an important question, doesn't it? If cannabis leaves, parts often just considered waste, can actually yield such rare and potentially valuable flavoalkaloids when we use these advanced analytical techniques. Well, what other so-called waste products out there? Across all of agriculture, natural ecosystems, think about fruit peels or spent grain from brewing, wood pulp maybe. What might they be harboring? Undiscovered compounds with significant medical, industrial, or environmental applications, just sitting there, waiting for the right scientific methods to reveal them. It really prompts us to fundamentally rethink our definitions of waste and value in the natural world. It suggests that true innovation often lies in, well, looking very carefully where maybe no one else thought to look before.
That is a fantastic, really thought-provoking idea to leave us with. Thank you for joining us on this deep dive. And we absolutely encourage all of you to keep exploring the hidden complexities of the world around us. There's always, always more to discover.